Now I'm going to shift to what's the status of developing cures and finding new drugs and the challenge that this poses for this country. So this table sort of highlights historically what the costs and timelines for developing drugs. And so if you look at the table, in 1970, it took 7 years and $140 million to get a drug approved and registered and launched and to take care, to provide it to physicians and treating patients. And since then, every decade, the costs and time to getting those drugs to market are staggering increasingly. So now it takes over $1.2 billion and 12.5 years to get a drug approved for market. And these all falls within the context that there's a 20-year patent life on a drug from the very beginning that is discovered to the point in which that patent life ends. So there is increasingly a shorter timeline for a pharmaceutical company to recoup its return on investment. Also, the drug development failure rate is extremely high. It's over 80%, and for cancer, it's over 90%, and for neurological disease, it's over 95%. So it's increasingly very difficult for the pharmaceutical companies to achieve a return on investment within their patent protection period. So if you think about that, these costs are really non-sustainable, and drug makers are becoming increasingly risk averse of really finding a cure, because basically a cure means no customer for the customer, and so they need to find a way to have an ongoing customer for that 8 years in which they have that continued patent protection. So there's obviously a disincentive for finding cures. So the only way that they typically will focus is on incremental healthcare by trying to control disease and alleviate symptoms. So one of the proposed solutions to turn this around is the use of stem cells, because stem cells have the capacity for regenerative medicine. They also could be used as tools to accelerate drug discovery. And so just to basically give you a sort of a layman's background of stem cell biology, stem cells have two properties. They have the ability for self-renewal, and they have the ability to differentiate into a variety of different specialized we call specialized cells or progenitor cells. Now, embryonic stem cells includes the area of cloning, and they can produce over 200 specialized cells, but obviously they have controversy surrounding them. They also, if you inject an embryonic stem cell into a mouse, they will form a tumor. So these are very genetically very unstable types of cells. Now, in contrast, there are other types of cells that we categorize as adult stem cells. These are stem cells that are derived from patients. These are derived from postnatal tissue like cord blood, the umbilical cord, the placenta. And there's another type of stem cell that I'll briefly talk about, which is called the inducible pluripotent stem cell, or the iPS cell. And lastly, there are good stem cells, there are bad stem cells, and there are cancer stem cells that actually are responsible for producing a tumor. So the latter three groups are stem cells that are supported and endorsed by the Catholic Church. Now, terminology, you will hear the word pluripotency, and every stem cell does not have the same capabilities. And basically, stem cells are divided into basically categories based on their ability to differentiate and form different types of tissues. So if you think about the earliest period of embryonic development, basically everything is derived from three primitive layers in the embryo. And we call it endodermal, mesodermal, ectodermal. And so in Latin, endo, inner, meso, middle, ecto, outer. And from those specific layers are derived the variety of different cells that are listed in these different categories. So something that is called unipotent can only produce one specific organ. Something that is called multipotent can perform differentiated cells from at least 2 categories of germ cell lines. And a pluripotent can produce all of these cells from all 3 germ lines. So that's where the terminology arises. So embryonic stem cells and iPS cells are the only type of cells that are considered pluripotent. But what about adult stem cells? Where does our country, our government, leadership lies in terms of supporting adult stem cell research? So these are statistics from the government. This is from the Congressional Research Services that tracks where the dollars are going from NIH to support various different types of stem cell research. And what I've done here is basically show over time from 2003 to 2011, which is the last reported statistics that I have, breaking down stem cell research into human embryonic, embryonic non-human, adult human, and non-adult human. And these are the annual percent growth rates of NIH funding. And you can see that embryonic stem cell research receives the highest rate of growth of funding from NIH, whereas human adult stem cell research receives the lowest level of annual growth rate in stem cell research funding. So by the year 2017, if you project these numbers, by 2017, NIH will fund in total dollars more towards embryonic stem cell research than adult stem cell research.